Hi there, just another video. This week we're looking at DNA and the production of proteins, and what I'm going to do here is just go through an example of one of the types of problem solving questions that can come up within this topic. So the question here says that there's a single strand of a DNA molecule which has 6,000 bases. Of these bases, 1,600 are adenine and 2,300 are cytosine. What the question wants us to work out is how many guanine bases would be present on the complementary strand of this DNA molecule. So it's important when you're reading through this question that you recognize a few things. The question originally talks about a single strand of a DNA molecule. You should know that DNA is a double-stranded molecule, so it's made up of two strands. This is telling us about one of these strands. So one of these strands has 6,000 bases. If one of the strands has 6,000 bases, the other strand also has to have 6,000 bases because they link up, the bases link up in pairs. You should know A links up to T and C links up to G. What the question is asking us to work out is how many bases, guanine bases in this instance, are on the complementary strand. So we need a little bit of background knowledge, as I said. We should know that DNA, double-stranded molecule. So this is the original strand that they're talking about in this top part. That contains 6,000 bases. We will also have in the DNA molecule a complementary strand that they're talking about here. So this has got 6,000 bases up the top, and this is going to have 6,000 bases at the bottom. These bases on the top can only be four things. They can either be adenine, thymine, guanine, or cytosine. If there's a base on the original strand that's an adenine, the base on the complementary strand that's going to pair up with this, we should know this from our base pairing rules, is going to have to be thymine. If there's a thymine on the original strand, the base that's going to be pairing up with this is going to have to be an adenine. If there's a guanine, it's going to have to be a cytosine, and if there's a cytosine, it's going to have to be a guanine. Okay, so we need that bit of knowledge about DNA molecules before we go on and carry out the problem solving exercise. So if we look back at the question, it tells us that in this original strand, 1,600 of these bases are adenine. So if there's 1,600 adenine bases, that means that on the original strand, that means that on the complementary strand, there's also going to need to be 1,600 thymine bases, because these adenine bases need a partner to link up with, and we remember adenine only pairs with thymine. So if there's 1,600 of these on the original strand, there's going to have to be 1,600 of these on the complementary strand. The question also tells us about cytosine. So cytosine, there's 2,300 cytosine on the original strand. Well, remember, cytosine links up with guanine, and if there's 2,300 cytosine on the original strand, that means that on the complementary strand, we're going to have 2,300 guanine. So that leads us to the answer of our question. Because there's 2,300 cytosine bases on the original strand, that means that 2,300 of the bases on the complementary strand are going to need to be guanine. Okay, hope that makes a little bit of sense to you. If you're having any difficulty with that, just send me a message on the comment section of Show My Homework and I'll give you a hand. Okay, thank you.